Hello, and welcome back to Introduction to Genetics and Evolution. In this set of videos, we'll be getting to the Hardy-Weinberg equilibrium. This is a foundational concept for the field of population. Before we go to that, however, let's talk about allele frequencies and genotype frequencies, because understanding what those are and how you get them is very important both for the Hardy-Weinberg equilibrium and for population genetics more broadly. Now let me ask a couple of broader questions first. First, can we predict genotype frequencies if we know allele frequencies? If we know how abundant different genotypes like big A, big A, big A, and little a are in a population, can we get from this to allele frequency? And if the answer is sometimes, when can we? The other question is, do genotype frequencies intrinsically change over time, or do they remain constant? Let's assume, for example, that big A is dominant over little a. Do we expect over time big A's to become more common just because of dominance? Well, we'll answer these by looking at variation one gene at a time. We're not going to be looking at interactions among genes here at first. So let's take this hypothetical scenario for the A gene. Let's say this A gene has two alleles, big A and little a. And let's say there's three possible genotypes, of course, from these two alleles, big A, little a, and little a. Well, by definition, frequencies have to add up to 100%. If there's two possibilities, as with the alleles, if there's a big A and a little a, the total of the fraction of little a plus the fraction of big A must be 100%. So if we have 78% big A, then we necessarily must have 22% little a. Same thing happens with the genotypes, that if 25% of individuals are big A, 50% are big A, the only other possibility is little a, and therefore that must also be 25%, making the total 100%. So how do we calculate genotype frequency? Well, we can definitely calculate genotype frequencies always when we have the counts of the different genotypes. So let's assume in this case that every individual is a diploid. They have two copies of all their genes. So we can literally just get this by counting. So here's a population. There's big A's, and little a's. There are 10 individuals in this population. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The frequency of big A will be its abundance, which there's only one right here. So it would be 1 out of 10, or 0.1, which is the same as 10%. Frequency of big A, little a's, there are 1, 2, 3, 4, 5, 6 out of 10, or 0.6. For little a's, the ones that are not circled, it's 3 out of 10, it's 0.3. So you add up these three numbers, and again, it adds up to 100%, as we expect. What about allele frequencies? Well, there's two ways we can actually do this. The most obvious way, which is not necessarily the better way, is to literally just count the big A's and the little a's in this group. So we can look at this and see, well, there's 20 total alleles here. You know, they're attached together as genotypes, but there are 20 alleles. How many big A's are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 big A's out of 20. So how many little a's are there? There obviously must be 12. So we have 8 out of 20, or 0.4 big A's, 12 out of 20, or 0.6 little a's. And again, this adds up to 100%. The better way, when we're working with a large number of genotypes, rather than counting the individual alleles like this, is to look at the genotype frequencies. We can say, we know that big A's are completely made up of the big A allele. So the frequency of big A would be the frequency of big A's, and half the frequency of big A little a. So these are the numbers we used from the previous slide. Remember that our frequency of big A was 0.1. Our frequency of big A little a is 0.6. This is from the previous slide. We add up 0.1 plus half of 0.6, which would be 0.3, comes to 0.4, which is exactly the same as what we saw up here. Similarly, we can say the frequency of little a, or 0.3 plus half of 0.6. So that's 0.3 plus 0.3, and that is 0.6. That's exactly what we see over here. And again, these add up to 100%. This is what we expect. Now, you can think of the world as basically a pool of gametes. It's not a particularly pleasant thought, but you can think of it that way. All individuals of sexual species start as two gametes, right? That you at one point in time were two different gametes that came together and fused into you. Now, gametes are one end. They just have one copy of all their genes. And this vision is not as artificial as it sounds if you just imagine, as it shows here in this picture, all the gametes floating around, because many marine invertebrate species spew their gametes into the water, and those are the ones that ultimately come together to make individuals. In this picture here, about 60% of the gametes are big A, 40% of the gametes are little a. And what we can do to estimate, if we assume that these gametes just come together completely randomly, we can use a joint probability multiplication to figure out what the genotype frequencies will be in that next generation, the people who are the individuals who came together from the gametes. So if 60% of the sperm are big A, 60% of the eggs are big A, so we're just saying 60% of all the gametes, 40% of the sperm are little a, 40% of the eggs are little a, what would the probability of a big A individual be? Well, the probability of a big A individual would be the joint probability that an A sperm comes together with an A egg. Well, A sperm is 0.6. A egg is also 0.6. So that would be 0.36 or 36%. We can do this for all possible individuals. So here's, 0.6 times 0.6 is 0.36. For little a, it's 0.4 times 0.4, so that's 0.16. But there's two different ways you can make a heterozygote, right? You can have a big A egg and a little a sperm, or a little a egg and a big A sperm. So each of those is 0.6 times 0.4, 0.6 times 0.4, or 0.24 twice. That comes out to 0.48. And again, as we expect, we can add all these up. It comes out to 1.0 or 100%. So we're assuming here that the gametes just come together completely randomly based on their proportions in the population, which is an assumption we'll come back to, and that's relevant to Hardy-Weinberg. Now let me show you just another way of visualizing the same thing. Rather than using this sort of joint probability approach, let's visualize it a little bit differently. We can use a modified Punnett square to show the relative amounts of the big A and little a gametes. So again, this is exactly the same thing as 0.6 big A, 0.4 little a. And we have here the sperm on this axis, the eggs on this axis. Note the size is here, the relative size. This is doing a 6x6 block. This is a 4x6 block, 4x6 block, and 4x4 block. We can use that then to estimate what we'd see in terms of the genotypes. And doing that, we see exactly the same results that we saw before. That here we have 36% of all these blocks, or 36 out of 100 blocks are big A, 16% or 16 out of 100 are little a, 24 plus 24 is 48% big A, little a. Now, can we calculate the allele frequencies from these genotype frequencies? Well, the answer is yes. We can always calculate allele frequencies when we have the genotype frequency. Because again, we know all the big A's have big A. They are all big A, so 0.36% is that. Big A little a's are half big A, so half of 0.48. So 0.36 plus 1/2 of 0.8 is 0.6. So here in the offspring, just like in the gametes that formed them, the frequency of big A is 0.6. Frequency of little a has to add up to 100%, so this must be 0.4. Okay? So we see that not only, so we have the frequencies of the alleles being 0.6 and 0.4, the genotypes being 36%, 16%, and 48%. And then in the next generation, if they were to spew out their alleles again, the allele frequencies are still the same. Since the allele frequencies are still the same, this is all a self-perpetuating process. That the allele frequency was 0.6 in the gametes, it created these genotype frequencies, and these genotypes will produce 0.6 frequency of big A gametes. So this is a process that is basically stable with these assumptions. Now the pattern that you see does have a wide variety of assumptions, but the stability is what's referred to as the Hardy-Weinberg equilibrium. We'll come back to this more in the next video. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about calculating allele frequencies, we talked about calculating genotype frequencies, and the important thing I emphasized at the very end of the last video was with these sorts of assumptions that gametes just come together at random based on their proportions, we end up with a stable equilibrium. And in essence, the allele frequencies don't change over time, the genotype frequencies don't change over time. That is, by definition, an equilibrium. Now this pattern was first described by these three gentlemen here. This is Godfrey Hardy over here, Wilhelm Weinberg over here, so they're the ones who typically are named for this as the Hardy-Weinberg equilibrium. There's also American William Castle introduced a similar idea in around 1903. But the important thing was this idea of self-perpetuation. And this is what I showed you in the end of the last video here, that you had an allele big A allele frequency of 0.6 in gametes, it created this set of genotypes with the little a allele. 0.36 were big A, 0.48 were big A, little a, 0.16 were little a. The following generation, you would again have this 0.6 frequency of the big A gametes, and correspondingly the 0.4 for little a. Now, up until 1902, several people thought that it was possible that the dominant alleles would intrinsically increase in the population. They are, after all, called dominant. And some people also assumed that rare alleles would just tend to get lost and have this inherent drive towards loss. Now, it was in 1908, and also partly from the 1903 contribution, Hardy and Weinberg independently showed that both these assumptions were not true. That if you can just use this purely probabilistic approach, allele and genotype frequencies will stay completely stable. Now, there are assumptions underlying this, and we're going to come back to those assumptions in just a moment. Before we come back to that, let's change this into more mathematical notation. So let's formalize the math. Now, let's say these are typical notations people use. Let's say the frequency of big A is referred to as P. Let's say the frequency of little a is referred to as Q. And again, we're assuming there's only two alleles in this population. Because there are only two types, P plus Q must necessarily equal 1. Thus, if P is 1, then Q must necessarily be 0. You can't have negative numbers because these are frequencies. What would the frequency of big A be? Well, it would be the probability of a big A encountering another big A. So necessarily it would be P times P, or P squared. Right? Well, we can push this in. P squared for big A, Q squared for little a, and 2PQ for big A, little a. Well, why is it 2PQ? Why isn't it just PQ? Because doesn't it just mean a big A and a little a? Again, there are two different ways you can have it. You can have a big A sperm and a little a egg, or a little a sperm and a big A egg. So there's two different ways you can get it. Again, these genotype frequencies must necessarily add up to 1. So P squared plus 2PQ plus Q squared equals 1. You note that this quantity squared will come out to that. Right? P plus Q squared is P squared plus 2PQ plus Q squared. So it all comes out very elegantly. Let me show you how the frequencies would look if you were to plot these things together with these assumptions. So what you see in this graph here, on the x-axis are the frequencies of big A and little a, P being the frequency of big A, Q being the frequency of little a. And on the y-axis here, you see the frequencies of the different genotypes that are depicted with the different colors here as well. So big A is, or P squared is in red, little a, or Q squared is in blue, green is 2PQ. Okay? And as you can see, the abundance of the heterozygote peaks out around 50%. And as you get to the two ends, you end up having one of the two homozygotes being completely abundant. Obviously, at the extreme end here, if there are no big A's, then everybody in the population must be little a. If there are no little a's, then everybody in the population must be big A. Now, this allows you to infer genotype frequencies from allele frequencies, because you can say, well, I know the allele frequency is 0.8 for big A and 0.2 for little a, so you can immediately say, oh, here's the abundance of big A, little a, little a. However, some assumptions have to be met, and we haven't gone into that yet. Now, let me give you 3 points. The third is going to come back to these assumptions. Number 1, you can always know genotype frequencies from genotype counts. And I showed you an illustration of that before, but here it is as an example. Here's big A's, big A, little a, little a. Well, all you have to do to figure out the genotype frequencies from the counts is add them up to find the total and divide by the total. So let's do that. Add them up. There are 200 total individuals. Divide each by 200, and there are your genotype frequencies. That's all it means. It's just the relative abundance of the 3 genotypes. And again, this always has to add up to 100%. Point number 2. You can always know allele frequencies from genotype frequencies. Here, let's say, for example, we have these 3 genotype frequencies. We can use this little trick I showed you before. All the homozygote plus half the heterozygote gives you the particular allele frequency. So, for example, for big A, all the homozygote is 0.04 plus half the heterozygote, which is half of 0.32 or 0.16, comes out to 0.20. All of this one's 0.64 plus half of 0.32, which is 0.16, equals 0.80. And again, they add up to 100%. The third one, though, the third point is you cannot always know genotype frequencies from allele frequencies. That's what it seemed like we could do with that figure I showed you earlier, but let me show you why. Let's say, for example, that the frequency of big A is 0.5 frequency of little a is 0.5. Simple case, right? Well, you could have 0.25, 0.5, and 0.25, right? That adds up to 100%. If you do all of these plus half of these, that is 50. That is 0.5. All of these plus half of these, that's 0.5. That's actually at the Hardy-Weinberg expectation. But you could also have other possibilities that don't match the Hardy-Weinberg expected abundances. You could have this. 0.45 plus half of 0.10 is still 0.5. This 0.45 plus half of 0.10 is still 0.5. You could even have this. What if you had no heterozygotes? Again, the allele frequencies here are 0.5 and 0.5. So the bottom line is you can always calculate allele frequencies from genotype frequencies. The reason for this is that the alleles are the ingredients of the genotypes, right? But you cannot always calculate genotype frequencies from the allele frequencies because genotypes are specific combinations of alleles. It's, again, just like ingredients. If somebody gave you a pancake, you could know it was made from flour and sugar and baking powder and things like that. In contrast, if somebody gave you flour and sugar and baking powder, there's a lot of different things you could make with that, right? Many combinations are possible. So coming back to Hardy-Weinberg, Hardy-Weinberg allows predictions of genotype frequencies from allele frequencies under certain conditions. And this is when that sort of simple multiplicative rule applies. Well, those conditions include random mating. This is where the multiplying probabilities rule works very well. We don't assume that big A's are more likely to encounter other big A's than little a's relative to the proportions in the population. It assumes no selection, migration, or mutation at the locus. In terms of selection, we're assuming that it's not like the big A individuals tend to die more readily and therefore are not constant. We assume an infinite population size. And this is why probabilistically everything works out very nicely. This is referring to the absence of what's called genetic drift. We'll come back to this in a later lecture. But overall, Hardy-Weinberg is predicting a completely boring population that probably could never exist. So why bother? Well, the answer is by seeing how populations deviate from Hardy-Weinberg expected frequencies, we can infer which specific interesting evolutionary processes may be operating. So we'll come back to this actually over the course of the next couple of lectures about some specific evolutionary processes that could be operating. But let's look a little bit at testing for Hardy-Weinberg. So here's an example. So here we have big A's are 245 individuals, big A, little a's 210 individuals, little a's 45 individuals. Let's walk through this one to see if this population is at Hardy-Weinberg, and then I'll have you do one later. So the 4 steps you want to do is figure out the true genotype frequencies, figure out the true allele frequencies, and then figure out the Hardy-Weinberg expected genotype frequencies, which may or may not be the same as the first one, and then ask yourself, do the true frequencies match the expected frequencies, or close enough to them? So let's do this now. Again, from the genotype counts, we can always get the genotype frequency. We just get the totals. 500, divide by the total, and here are all the genotype counts. So that's fine. These are the true genotype frequencies. From these true frequencies, we can get the true allele frequencies. So all of these plus half of these for big A, all of these plus half of these for little a. Very simple, 0.7, 0.3. Now the question is, if we put these together in the Hardy-Weinberg expected proportions, does it actually work out as we expect? Is it p squared for big A, 2pq for big A, little a, and q squared for little a? The answer is p squared is 0.7 squared, 0.49. That matches beautifully. 0.42 to 0.42 matches beautifully. 0.09 to 0.09 matches beautifully. So yes, this population is at Hardy-Weinberg. Now let me ask you to try this next one. See if this one is at Hardy-Weinberg. Try it out yourself using the same processes we just did. Well, thank you. I hope that wasn't too hard. So again, what we do is first step we do is figure out the total. So in this case, the total adds up to 400 plus 200 plus 400. That is 1,000. Nice, easy number there for you to start with. So the genotype frequencies, the true genotype frequencies in this case, would be 400 divided by 1,000. So that would be 0.40. 200 divided by 1,000, 0.20. 400 divided by 1,000, 0.40. Okay, so these are our true genotype frequencies. What about our allele frequencies? The allele frequency for big A would be all of these plus half of these. So that would be 0.4 plus 1/2 of 2 is equal to 0.5. Q sub little a is equal to same thing exactly, 0.5. You can check here at this point. Yes, they still have to 100%. These still have to 100%. So the question is then, do the Hardy-Weinberg expected genotype frequencies match these? Well, our expectation for big A is p squared, which would be 0.25. For big A little a, it should be 2pq, which is equal to 0.5. Little a should be q squared. Q is 0.5, so that would be 0.25. Uh-oh, these don't match up at all. 0.25 is not the same as 0.40. 0.5 is not the same as 0.20. 0.25 is not the same as 0.4. So people often tend to think this is circular, but as you can see in this case, it was not actually circular. We actually are seeing a deviation from the idea that these gametes just come together at random, everybody survives, and all moves on. So there is a deviation from Hardy-Weinberg in this case. Now, seeing how natural populations deviate from Hardy-Weinberg expected frequencies, we can actually infer what evolutionary forces are operating. In that previous slide, you saw a deficit of heterozygotes. There were fewer heterozygotes than we expected from Hardy-Weinberg. What does that mean in particular? Well, we'll look at that as one possible deviation in the next video. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. We've been talking about the Hardy-Weinberg equilibrium and how, under certain conditions, and only under those conditions, you can calculate or estimate expected genotype frequencies from observed allele frequencies. The opposite is always true. You can always know allele frequencies from genotype frequencies, but you cannot always infer genotype frequencies from allele frequencies. Now, in the last example I showed you in the last video, we saw a case where there were fewer heterozygotes, fewer of the big A little a individuals, observed relative to what was expected from Hardy-Weinberg. Now, why might that be? Well, this is going to be the first of many possible deviations from Hardy-Weinberg that we'll discuss, and it could be what's referred to often as the Volland effect. Well, let's look at a little bit of real data. Here's some real data from a Navajo population, looking at the MN blood groups. MN, just like big A and little a, there's two alleles, M and N. There's three possible genotypes, MM, MN, and NN. So let's take a look at what we see when we look at the Navajo at this particular group. Well, we can do the same test for Hardy-Weinberg we've done before. We figure out the total number of individuals, 361 in this case, get the genotype frequencies, the true observed genotype frequencies. From them, say all of these plus half of these, and there are allele frequencies. So our allele frequency for big M is 0.917, for big N it's 0.083. Take this squared, 0.841, 2pq, so 2 times this, 0.152, 0.083 squared is 0.007. Now we see this population is not absolutely at Hardy-Weinberg, but it's very close. It's very close to the Hardy-Weinberg predicted frequencies. So let's look at another population now. Let's look at these Aborigine. Now again, if we look at the MN blood group in these individuals, we follow the same procedure. I won't go through all the steps, but if you want some practice, you can pause the slide, take these first set of numbers and go through and do it yourself. We come back to, again, a set of genotype frequencies that are predicted. They're very close to those observed. 0.031 is very close to 0.030, 0.293 versus 0.296, 0.676 versus 0.674. It's within 0.003 for all of these things of the expected genotype frequencies. Now let me ask you a funny question. What if you were an alien from another planet, you came down and you grabbed a bunch of Aborigine and you grabbed a bunch of Navajo separately, but you put them all together into the loading bay of your spaceship. And you have this mixed population. So let's say it's exactly the same individuals who are genotyped. Now we saw the Aborigine were at Hardy-Weinberg, the Navajo were at Hardy-Weinberg. What happens when we put all these together? Well, we get these allele frequencies for M and N, but we get something that's deviating rather dramatically from Hardy-Weinberg expectations at the genotype frequencies. Look at that. This is dramatically different. This is dramatically different in this one as well. You notice especially, and this is one I want to point out in particular, we expect almost half the individuals to be heterozygote, we observe only about a quarter of individuals are heterozygote. So there's a dramatic deficit of heterozygotes in the observed relative to the expected. That's like the last example from the last video. Now, why might we see this? Why do we see this deviation? We have a Hardy-Weinberg population, another Hardy-Weinberg population. Why isn't we put them together? That's not a Hardy-Weinberg. Like, what assumption have we deviated from? Well, one big assumption we deviated from the list I showed you earlier was the assumption of random mating. That the idea is that any two individuals are as likely to breed as any other two individuals. Remember from the first video from the series, the gametes just floating all around. It's not like that there, because the Navajo lady is not as likely to breed with an Aborigine as she is with another Navajo. So imagine that in one population, big A is abundant, then big A's are going to be very likely to encounter other big A's. In another population, little A's are very abundant. Little A's are going to be very likely to encounter other little A's. But big A's and little A's are very unlikely to encounter each other. And that's why you see this deficit of heterozygotes. And in this regard, the Hardy-Weinberg assumption was violated. That Hardy-Weinberg assumption was not rejected within the Navajo or within the Aborigine, but it deviates from this combined population. This results from non-random mating. Importantly, this will very typically result in having too few heterozygotes. You expected about half, you observed about a quarter. This pattern is referred to as the Volland effect. This is when you sample across populations. So the populations within each population may have random mating. But when you sample across or between populations, you get an underrepresentation of heterozygotes relative to Hardy-Weinberg. So this is a way for potentially identifying different populations. You can see how different or how much of this deviation you see. We'll use that actually in a subsequent video for calculations I refer to as FST. But let me ask you a different question first. Why does it matter? Why does it matter if something's at Hardy-Weinberg or not? Well, in fact, the first step in genome-wide association studies for genetic diseases or any traits is or should be to test for Hardy-Weinberg. Now, why is that? Well, actually, the genome-wide association studies assume Hardy-Weinberg is true, or assume that you're very, very close to it. Basically, you're assuming that there's linkage disequilibrium caused by close proximity between marker alleles and disease-causing alleles. Remember that fundamental purpose of all genetic mappings to see an association between genotype and phenotype. And we're hoping this association is from close proximity or lack of recombination. So imagine that you see something like where 20% of individuals with big A genotypes have a disease, and 5% of individuals with a little a genotype have a disease. Then we're assuming there's an association between the big A allele, or big A genotype, or the A marker gene more broadly, and the disease. But just being in different populations also causes linkage disequilibrium. Let me give you an extreme example to illustrate this point. Let's imagine that in population 1, every individual is big A. Okay? Just that simple. Imagine population 2, every individual is little a. And let's say that a disease is a little bit more abundant in population 1 than in population 2. Okay? Would you say that big A individuals are more likely to have a disease than little a individuals? The answer is yes, you would say this, because just the way it laid it out. Now, this is actually a fake LD between the disease and the gene, because the disease may not be on the same chromosome as this A gene in particular, and in fact, the disease may not even be genetic. Let's say, for example, in population 1, everybody eats a lot. In population 2, everybody has very good weight. You may see obesity is much more abundant in population 1 than population 2, but it may have nothing to do with your genotype at the A gene. So it's really important that you have true Hardy-Weinberg in doing these genome-wide association studies. Otherwise, the associations you see may have nothing to do with the genotypes you're observing, and the disease, in fact, may not even be genetic at all. Now, the punchline is if there are allele frequency differences between populations at a SNP, which is very often true, let's say the SNP is being used as a marker, and if disease incidence differences exist between the two populations you're studying, which again, very often true, sometimes for genetic reasons, sometimes for not, but it may not have anything to do with the particular marker or anything near that marker you're looking at, then a genome-wide association study will erroneously make it seem that a gene near the SNP is causing or contributing to the disease. Now, if you test for Hardy-Weinberg, you can avoid this error because you can identify if you're looking at one interbreeding population or not. Your hope is that the population is at Hardy-Weinberg or is very, very, very close to being at Hardy-Weinberg. And then if you see an association, you know it's not this weird bias. Now, although it's very important to test for Hardy-Weinberg, this is often not done. Here are excerpts from 2 studies from not too long ago. This is from the American Journal of Epidemiology in 2006. The exclusion of studies in which Hardy-Weinberg was violated changed the conclusions and changed statistical significance of gene disease associations. That's scary. Think about it. Millions of dollars go into finding these gene disease associations. We really need to be careful to know that we're doing them right. Here's something from the European Journal of Human Genetics in 2005. Testing and reporting for Hardy-Weinberg equilibrium is often neglected, and deviations are rarely admitted in the published reports. So this is a really big deal. There's other issues about interpreting deviations from Hardy-Weinberg. Let me show you this example here. So this is a real example where a Hardy-Weinberg test was done but interpreted incorrectly. This is raw data from a 2000 study of BRCA2 variants. These are from newborn males from a hospital in the United Kingdom. Just as a little test here, I want you to do the math for this and figure out how close this is to Hardy-Weinberg expectations, and do you see a particular kind of deviation? So try that out. I hope that wasn't too hard. Let me go ahead and show you the answers. These are the numbers you should have come up with. So these are the true genotype frequencies. These are the true allele frequencies. These are the Hardy-Weinberg expected genotype frequencies. What we see here is our expected frequency of the heterozygote is 0.4. Our observed was 0.36. So there is, or there at least seems to be, some slight deviation from Hardy-Weinberg and in this direction of too few heterozygotes. And this was, by the way, statistically significant too. Interestingly, how did the authors interpret this? The authors of the study interpreted it as that the big Allele individuals are less healthy than big A, or little a. They postulated that maybe there was some disease or there was some problem associated with big A individuals. In fact, there's a much simpler explanation that we're looking at newborn males in a hospital in the United Kingdom. It's quite likely, imagine this is a hospital in a place like London. It's quite likely that in a place like London, there's a lot of subdivision of the population, that people of, say, Indian descent are probably more likely to have kids with others of Indian descent. People from the Far East may be more likely to have kids with people from the Far East. People who are of European descent are probably more likely to have kids with others of European descent. Yes, there are cases where people will have kids with people from other ethnic groups, but by having this tendency there overall in the population, which undoubtedly exists, you will get exactly this pattern. It's basically the simpler explanation than that the big A little a individuals are less healthy is that there's a little bit of a Volland effect there. But that wasn't considered. They probably didn't take our PopGen class. Now, let me close with a little ironic tidbit. This is a quote from Hardy's 1940 book. This is when he was about 62. His book is called A Mathematician's Apology. I definitely recommend it. It's a very interesting read about the elegance and beauty of math. He said, "I have never done anything useful. No discovery of mine has made or is likely to make, directly or indirectly, for good or ill, the least difference to the amenity of the world." Well, I'd like to say very strongly, he was very wrong in this, that this Hardy-Weinberg idea, which he helped bring about and he helped popularize, really has made a huge impact. And we're continuing to see it now, even literally more than 100 years after the original publications of the Hardy-Weinberg work. We still see these applications for things like genome-wide association studies, the kinds of things he never would have imagined. So this is really cool stuff. Even though this was written when he was 62 and he was lamenting the waning of his mathematical ability, he really did a lot for the world, as did, of course, Weinberg and Castle. Thank you.